Our next speaker, continuing the vaccine theme, and from Jamie's group, is Claudio. Claudio.
Thank you. I'm Claudio. I work with Jamie in Jamie's lab. I'm a postdoc. Today I'm going to give you an update on our vaccine program. So yeah, this is the pipeline of the TB vaccines. As you can see, there are in the pipeline different kind of vaccine encompassing live vaccine, whole cell, viral vectors, and subunit vaccines. And they are all on different phase of clinical development. Having seen the recent development of the COVID vaccine, you can see that there is mRNA are missing, so there is opportunity to develop something in that space. But however, what we've been working on is subunit vaccine protein in adjuvant, and with each subunit vaccine is composed of an antigen and an adjuvant, and this is our antigen, which is a multi-stage antigen composed of a fusion protein of two tuberculosis antigen FIB and CISD. Antigen FIB is a most famous antigen of Mycobacterium tuberculosis. Is the most abundant secreted protein, and CISD instead it's a enzyme that catalyzes the absorption of sulfur from the extracellular environment, and it's necessary for M. tuberculosis to build proteins that contains methionines and also to resist to the intracellular environment of the phagolysosome. We've made it a fusion protein, so it can kind of act, the antigen FIB part acts on the early stages of infection, whereas the CISD part acts on the later stages of infection. And as you can see in the right-hand side, CISD is lower expressing at the beginning of the infection in the lung, but then it gets upregulated, whereas antigen FIB, on the other hand, is highly expressed at the beginning and gets downregulated. We combine it with an adjuvant called Advax, which is a proprietary of a biotech company in Adelaide, Vaccine. It's a novel adjuvant. It's made of inulin, which it's a polysaccharide derived from a plant. And in its delta form, it forms those little donut-shaped particle of 2 micron in diameter. So it's very similar to a bacteria. It's been used in clinical trials in humans, and it's been proven to be safe, and last year has also been used in clinical trial for COVID vaccines. It's known in animal models to produce Th1, Th2 responses, and these responses have been shown to be effective against influenza, HBV, and other pathogens. We combine it together, TB-CRE with Advax in our formulation, and we showed that given intramuscularly was as effective as BCG in the Black 6 model in inducing protection and in reducing the pathology in the lung of infected mice. But then we moved on to mucosal vaccine, thinking that by inducing a response in the lung, we could better, we had T cells and immune response in the lung where the actual infection takes place. And we compared the vaccination to the one induced by the intramuscular route. And in this section here, in this graph, you can see that the Advax CpG intratracheally was able to induce a protection that was greater than the one given intramuscularly. And when we looked at the immune response after infection, one of the main striking difference was while the TB-CRE Advax CpG intramuscularly was able to induce a multifunctional gamma, TNF, and IL-2 response, the main difference was that the intratracheal one was able to induce the same response with the addition of IL-17. So taking a step back and looking at the induction of that immune response and immune cell recruitment to the lung, for 4 weeks after vaccination, we looked at the CD4 compartment in the lung and looked if there was actually an increased population of tissue-resident memory cells. And we used all these markers to determine tissue-resident memory. And we could see that in the TB-CRE Advax intratracheal group, there was a total CD4 expressed higher level of markers that were indicative of tissue-resident memory. And when we looked at the antigen-specific cells using a tetramer marker that is able to bind only the CD4 T cells that are specific for the antigen 85B, we were able to see that these T cells were upregulated in the intratracheal vaccinated mice and were lasting up to 8 weeks after vaccination. And all these cells were highly expressing markers of tissue-resident memory. The cytokines that they were expressing, again, were IL-17, IL-2, and TNF. And if you look at the green side, the antigen 5B tetramer positive cells, they were highly enriched in IL-17 and ROR gamma T, which is the master regulator of Th17 differentiation. So we decided to have a look at what is the role of this IL-17, and we tried using monoclonal antibodies that was able to block the activity of IL-17, and we did that 1 day before infection and throughout the infection after vaccination. And as you can see from this graph, while anti-IL-17 didn't have any effect on the unvaccinated mice, the level of infection was similar, while in the CISVAC2 vaccinated mice that received an isotype control, there was still protection, whereas the one that received the anti-IL-17 treatment lost completely protection. And in parallel with that, we saw that the IL-17 treatment reduced the recruitment of neutrophil to the lung, as well as the accumulation of CD4 T cell specific cells. So that's why I think that the IL-17 is doing in this setting. But how it actually Advax and Adjuvant work? There has been 2 brilliant PhD students in our lab that went on and tried to work out the mechanism of action of Advax, and by giving it intratracheally, they were able to show that after 3 days after instillation, Advax particles in green were able to be distributed throughout the lung evenly and uptaken by CD11C positive cells. And as you can see, all these little dots are the little donut-shaped Advax particle. And then they used flow cytometry to monitor the level of a wide variety of antigen-presenting cells, and they were all coming in and upregulating and expressing several activation markers. They did that by using mass cytometry, which uses 40 different markers to define the population and to determine their activation state. And as you can see in these graphs, there were several population in the innate at the top or in the adaptive cells at the bottom. There were several markers that were upregulated. And they published this earlier this year in Mucosal Immunology. In recent development of this, Erika Stewart kept going on to try to identify the molecular basis by which Advax works, and in collaboration with the Institute of Glycomics at Griffith University, they used the glycan arrays that try to manage and identify the residue on Advax and what type of lectins are able to bind the adjuvant. And as you can see, there were several hits that were determined that there was some residue of manosyl, of GalNAc, and of GlucNAc that were able to, that are present probably on the surface of Advax. They are mediating the uptake and the activation of the innate cells. And these residues are in the mice receptor are kind of be able to be bound by several receptors such as DC sign, DCR, and lectin-1. And so these are now going to be investigated if they are actually the one responsible for the Advax activity. So in conclusion, putting all this data together in a chronological order of what's happening, we think that Advax possibly bind one or more lectin on cell surface. It gets internalized by IPC across the lung by a wide variety of them. It induces a broad cellular recruitment and activation, which in turn activates specific Th1, Th17, CD4 positive T cells that express markers of tissue residency, and induces a protection that is enhanced compared to the parenteral intramuscular administration, and an IL-17 response, which is essential for the protection against MTB challenge. And I'd like to acknowledge everyone from Trika's lab and Warwick's lab and our collaborators. And thank you very much. I'm ready for questions.
Thanks, Claudio. If there's any questions for Claudio on his work, yeah, this. Just want to see if we have a room now.
I'll bring it back for you. So, nice work. So when you put the vaccine intratrachely, do you get lesions in the lung because of the vaccination?
So we haven't seen any lesions. No, we've only looked after infection, so it's been kind of further away. But after infection, the lung is clear. I can see the lesions in the unvaccinated mice,
Right.
but the vaccinated mice don't have any granulomas or any particular lesion at that timing point. But Erika is going to look at the effect on the lung physiology if at early time points and compare it to other adjuvant and see if there is an impairment in lung physiology after the intratracheal vaccination.
So why would you think that just giving it from a different route would actually induce IL-17? As in, if you give it intramuscularly, you don't get IL-17, but
That's
if you
correct.
give it intratrachely, you do. So why would you suspect that?
We think that the lung is a kind of a different environment, and it's prone to induce a different response. For instance, there is more production of TGF-beta, which is a cytokine that is inducing more Th17 polarization in the lung compared to other sites.
Then you're saying that you don't think from an IM the actual vaccine itself, the protein and the adjuvant get to the lung?
No, from
It
the IM,
doesn't?
I don't think so. No.
Oh, okay.
I think BCG does.
Yeah,
we've
BCG
seen,
does.
We've seen that BCG is alive, but we haven't seen any evidence of that.
Michaela, last question quickly. We'll wrap
Thanks.
up.
Claudio, in humans, is the production of IL-17 associated with better outcomes for TB infection?
So there has been some vaccines that induce IL-17, and they've been shown to have a sort of a protective signature. There are also different kinds of IL-17. Some IL-17s are considered to be too inflammatory and bad, but there is no evidence in humans at the moment that that's the case.
Thanks, Claudio.
Thank you, everyone.